Thank you very much. We can, for the interest of time, we'll just get right to the presentation, but a brief 30 seconds of my history with Bangkok and Chulalongkorn, as well as the new emerging infectious disease centers. I'm very fond of this team and this research community because I originally trained here as a young scientist in 1987 and 1988 at the Thai Red Cross. So it's been quite a number of decades, and I continue to come back to Bangkok to learn from my colleagues here at Chulalongkorn, Mahidol, and many of the other medical centers. When I was asked to give this presentation, I thought it would be useful to share the lessons learned from countries and organizations that have made mistakes while they've done really impressive things. So we thought it would be good to show some of the successes as the first project of how we made it a rapid vaccine using the new mRNA technology in the US government, but also to point out how we overlooked some things. As virologists and infectious disease doctors, we actually knew better. And if we were to do it all over, we would make vaccines every bit as fast, but we would make vaccines that prevented the escape mutants and would not allow our vaccinated patients to infect others. As many of you know, the vaccines do not prevent viral shedding. So I'm giving a nod to the Thai Red Cross Emerging Infectious Disease Center here, as well as the One Health program. It's called Texas A&M. It's the middle logo at the bottom, which is in 16 countries working on One Health issues, much like the work that's being done in the Thai Red Cross and the One Health program here. Third, I'm a doctor at Massachusetts General Hospital, where I deployed to disease outbreaks. And lastly, I work with Rescue Medicine, which is a disaster organization. Next slide, please. Okay. There you go. The disclosures are straightforward. There's no financial interest conflicts. And if I talk about the use of medicines or therapies that are not for their desired indication, I will tell you that. And thirdly is, although I am a former US government official and infectious disease doctor for the government, the position and statements that I share are not the position of the US government. Next slide, please. Great, thank you. So here's where we're going in our short time together. And we're setting this up to support a dialogue in our panel later this morning. But we'll first look at the lessons learned from programs that have moved very quickly to make the world's first COVID vaccines, starting with Sputnik in Russia and the Pfizer vaccine, followed by Moderna. And then we'll also discuss briefly several of the Chinese vaccines for their strengths and their non-strengths. We'll then look at the body of our presentation, which is the science part, and we'll be looking at variant-proof vaccines. Are they even possible? How do we, in a year from now, when we're dealing with new variants of Omicron after BA.2, how do we prevent the new vaccines and use them in a way and design them in a way that prevents BA.3 and BA.4? And we are running out of Greek letters in the alphabet. We are already on Omicron. We only have 7 more letters in the Greek alphabet to name new viruses. So maybe that's another example of why we should bring this pandemic to a close. And lastly, we'll spend a couple minutes on the lessons that have been introduced to the global community from international collaborations, including with some difficult partners in difficult countries, but where we can get along by working on common principles, on things we all agree with. And as long as we work faithfully, transparently, and with respect, then we can accomplish a lot, even if the governments of these countries don't get along. In the bottom of this slide are some references which all hold the footnotes of many of the concepts I'm talking about. And you can click on them after this talk, or if you're really bored on your flight home. Okay, next slide. Okay, so section 1 is to focus on a really remarkable project to develop emergency vaccines against a virus from which we had very little history with. Traditionally, vaccines take 11 to 14 years to produce. Prior to COVID-19, the fastest vaccine that was ever produced was the mumps vaccine in the mid-70s, which took 4 years. And although we know that the science to make vaccines can move very quickly, the regulatory and the manufacturing and the clinical trials are 3 things that are moved very slowly. So it was very extraordinary in February of 2020 to sit in Washington, D.C. and be challenged to make a vaccine in 200 days by the fall of that year. And so we're going to share some wins and some losses on that journey. So if you find yourself in the position of working with a company or working with your government, hopefully you'll learn from our mistakes. Next slide. All right, so many of us were in China at the time of the outbreak. Because of a remarkable relationship with my hospital and 3 hospitals in China, American infectious disease doctors were already in China after January 2nd of 2020, working with their colleagues that they had previously worked with on bird flu outbreaks and SARS-1 in Hong Kong and Guangzhou. And we were just working on flu programs when all of a sudden we heard about an ILI, an influenza-like illness, up in Wuhan and Hubei province, which was already discussed. So moving quickly through those early days, it's important to know that our colleagues in Wuhan, these were friends of the Chinese doctors we were working with in Nanjing, they reported to us a couple things. The 3 bullets, the 3 discoveries that they shared at their peril, but which helped the rest of the world to prepare, was the documentation of person-to-person spread. 4 nurses in Central Hospital of Wuhan and the Chinese Traditional Hospital had become infected after being in contact with patients that they were caring for with this new infection. And they went on to infect people in their homes. So literally by January 9th of 2020, with barely 10 days of actual patients in the hospital, we had confirmed through well-trained Chinese investigators that person-to-person transmission was likely with this virus. The second point was the horrible finding that the R0, or the infectivity rate, was somewhere between 3 and 6, meaning one person can infect as little as 3 or as many as 6 people as a primary contact. And thirdly, in a small group of only 28 patients, 17 of them were made up of elderly Chinese urbanites from Wuhan, and they were doing terribly. They had high blood pressure. Several of them had COPD from chronic tobacco usage and diabetes in the 3 cases, and each of them had severity scores that were so far in advance to the younger patients. So early on, these examples were a warning sign to us that this was a rapidly moving virus. It was a dangerous virus. It would spread very quickly, and it would significantly impact elderly populations. Down below, only 3.5 weeks later, was the second experience that we had on the Diamond Princess cruise ship, which had picked up a number of Chinese nationals in Hong Kong and sailed through South China and developed a number of cases very early in the middle of January. Now, the reason why the Diamond Princess was so important is it was largely a closed environment. It was a laboratory to study viral infection among crew members who would sleep 8 to a room, and these very nice cabins which had good air ventilation to prevent infections. However, we compared these 2 communities, and that helped to prove to us that the virus had an incubation period as close as 2 days, that asymptomatic spread was possible, that people that were isolated in their cabin for 7 to 9 days, that never left their cabin, but one person would infect the second person and they would become symptomatic, but they were both infected. And lastly, because the manifest of the Diamond Princess showed nearly 1,000 passengers had traveled from the ship to 17 countries, we were certain it was going to be global. My point is that the experiences of our international partners and the experiences from Wuhan, from well-trained physicians in this strange experiment, if you will, of the Diamond Princess cruise ship told us we were in for a big problem. Next slide, please. But early warnings can come from our colleagues too. And I will start with this presentation the way that I'll end it, with the importance of even, fair international communication, collaborations with colleagues that we value and trust and work closely with over the years. In the top slide is a picture of Nian Jianwei, who is a long-term colleague who's in Wuhan, who we originally trained in Boston back in the 80s. So it was his work that helped us to learn that and he took a lot of risks telling the world about viral transmission. And on January 6th, he was one of the first persons to confirm airborne transmission by talking about infections of people that were many meters apart, although those people were not wearing masks at the time. And down below, a nod to Chulalongkorn and the awesome coronavirus research that's been done there by Dr. Tu and by the rest of her team. Her team is getting quite large now at the Thai Red Cross. Just pointing out that as early as January 15th, the first diagnosis of coronavirus outside of China was done here in Bangkok at Chulalongkorn in that laboratory. And lastly, it made use of not just the hard work of Chu and her staff and her team, but also the fact that they had been working on coronaviruses for quite some time. And it was that team that taught me how to get viruses from bats in Thailand many years ago. So thank you, Chu. Okay, next slide. Okay, so where are we at the beginning of an outbreak and how did an emergency vaccine program get underway? Presented here on the right side of your screen are the problems of an RNA virus that is a zoonotic. It's first in humans. For the first time, the virus is spilled over into humans and it's being tested by a brand new immune system and brand new populations that it's never seen before. So it undergoes dramatic increases in its mutation rate. If it was to jump into, say, a white laboratory mouse like a BALB-C or C57 Black 6, you would have very narrow evolutionary pressures on that virus. But when it's exposed to the elderly and the young, the diabetic, the HIV infected, the immune suppressed, those with malignancies, it tremendously expands the mutation potential and leading to multiple opportunities for fitness. So on the right side, how do you counter that? You must make sure that your vaccines go after epitopes that don't change this much, that will be with that virus through its phylogeny. So you got to find targets that will not mutate. That was mistake number one for the American program Warp Speed. Next point, the viruses might be local to the upper respiratory system and the lower respiratory tract leading to pneumonias, or they might be systemic, as is typical with several of the more dangerous influenzas. So your immunity must be local, or it might need to be systemic, as shown in the second row. We won't go through all of these, but I want to point out number 3. Row number 3, we knew the viruses came from animals, be it palm civet, raccoon, dogs, or one of the other mustelids or other animals on mainland China. But what's also interesting is we thought originally the virus made a one-way journey to humans and then took off. And it was quite disturbing when research in Southeast Asia and in the Americas proved that the virus would go back and forth from animal to human. And there's multiple examples where that back and forth travel produced new mutations for the virus that allowed it to become ever more variable and to work around our monoclonal antibodies and around our vaccines. So we'll stop with the rest of these. I'm just including them for completeness. Next slide. So the timing of a vaccine needs to be viewed in the context of how quickly you can move and make a vaccine, how quickly you can manufacture it for clinical trial, and how quickly the clinical trial can be done to produce a power analysis to tell you that it's not just safe, but it's also effective. But as we were sitting in Washington, D.C. in February of 2020, we said, should we come out with a quick vaccine in 100 days, or should we shoot for a very strong vaccine in 250 days? We tried to do both, but the early vaccines were not that good, okay? But what we're sharing with you is the modeling. If we were to come out with a vaccine very early in the pandemic, such as vaccine A, and we only could produce 100 doses of it, and it was only, let's say, 65% efficacious, that still would have helped a country. By targeting the elderly and by vaccinating healthcare workers and those with immune suppression, such as malignancy, organ transplant, and HIV, you could have potentially reduced the expansion and bought yourself more time. So should you do an early vaccine, or should you wait to try to do a later, more perfect vaccine? And the answer is undoubtedly that an early vaccine that is safe, that has some degree of efficacy, if correctly targeted, is going to dramatically assist your non-pharmaceutical interventions, your quarantines, your closures, your closings of schools, and your protection of vulnerable populations like the elderly. So as you think about this, understand that different viruses will make you move around on this chart depending on how severe the outbreak is. If it's a highly contagious flu that kills 40% of everybody, such as many of the H7 and H5 flus, you would want to go to battle with the vaccine that you have as early as you have it, and you would accept more risk. However, if you're dealing with a virus that moves more slowly and has a much lower case fatality rate, and you only have a certain amount of resources to build a vaccine, you might be waiting for that more perfect vaccine. Okay, so this is for all pandemics, and whether you're at COVID on one side or you're with a much more less lethal disease on the other side, we adjust the model and the prioritization of the vaccines when we select our vaccine candidates. Next slide, please. Okay, so here we thought we'd take you to the actual grading system that we had in May of 2020, so you can see our thinking as we looked ahead to predict which vaccines would be the most valuable. So the first point we'll make is that you see that eggs are in the bottom left hand. They are both likely to have low probability of success against coronaviruses. They've never been able to grow very well. You cannot grow coronaviruses very well in eggs. And they were unlikely to have high impact because there were no eggs to make a vaccine to cover the Western world and for exportation to the South and to the East. And if you look at DNA vaccines would have been pretty good on impact, but unfortunately, the probability of success was very low because the technology was very immature. mRNA, such as Pfizer, BioNTech, and Moderna, two groups that we had funded for years in the US, had a pretty reasonable chance. If they could do it, which was only moderate probability, about 60% probability, it would be very high impact. That's why it's high up, but not so far to your right. Other ones on there are interesting, and we'll call your attention to the plant-based vaccines. Yes, we're talking about plants like tobacco, which receive the gene for spike protein and grow extraordinary amounts of pure spike protein that can then be purified into a vaccine. And Medicago, a Canadian company, as well as several companies in Japan and Korea, are producing large volumes of new vaccines, which will help Asia's supply for the next pandemic. So very exciting stuff. Okay, so that's how we were thinking back then, and we got it mostly right. The mRNAs did better than we thought, and Pfizer-BioNTech was the first one to get an approved vaccine in the US FDA. Next slide. Okay, thanks for that. So Operation Warp Speed, if we are truthful to ourselves and we look back on it for our lessons learned, we would find that we stopped doing surveillance after we had the parent strain Wuhan 1 and after the British strain Beta, also known as BK117. Those were our two viruses. And once we had those viruses, we stopped looking. And we did not see the onset of Delta and Epsilon as they started to emerge, even though if we looked, we would have found them early. We would have found Delta within 8 months of the pandemic in northern Kerala in India, and we would have found Manaus in Brazil and Amazonia at the 9th month also of the pandemic. So by failing to look at your surveillance, we failed to correct our vaccines and make them more suitable for the variants. That was a big mistake. We noticed that an early vaccine would save more lives than a delayed vaccine, but we still got out there a little bit slow, and we still need to work on cutting more time. We believe we can make these vaccines in 100 days from the time that the virus sequence is now posted. We haven't done it yet, but we think that those are within our capability. In order for those vaccines to be useful, though, they can't just stop the infection in your country, in your community. They have to prevent variants from being selected internationally in other countries. So your vaccines must be easy to manufacture, easy to store without requiring crazy logistics like minus 165 degrees, and they must be low cost. That is not a statement on just world equality, it's a statement on practicality. If you make a low-cost vaccine, a bunch of people will add 25 cents to it or 5 baht to it so that it'll be profitable, and that will create an additional excitement in the industry and drive those vaccines into all countries of the world. The two red sentences here are very important. These are the biggest failures of our program in the US. The first one, the vaccine should prevent virus shedding. So once you're vaccinated, you cannot vaccinate, hurt, or infect another person. That did not happen. And lastly, they should prevent escape mutants. Next slide. Okay. So with that, what can we do to use the new information from 25 months of COVID research to help us to do better, not just for this pandemic, to help drive it to an endemic, but also for the next coronavirus pandemic, which might be very different from this one. And lastly, other respiratory viruses that are contagious and prone to mutations. How do we make it a one-wave pandemic, not a 5-wave pandemic like we're currently dealing with? So let's spend a couple minutes on that. Next slide, please. The first thing to point out is that we all live on the same world and that our borders are flat. If we put all sorts of restrictions on one animal population, say in the United States, how are we going to control migratory birds that fly from South America into the US? And how can the US protect itself, for example, from a duck that flies from Korea all the way up the East Chinese coast through Russia and then travels over 40 miles of ocean and lands in Alaska and infects the lady duck in Alaska, which then delivers viruses down our coast. So even though the origins of the viruses for influenza, for example, might be 12,000 miles away, in the polar north, they're only 40 miles away. So the world is much smaller for migratory viruses that travel on the wings of migratory birds, for example. This slide is stylized on influenza, and it looks at the migration of viruses through reservoir species such as wild birds into ducks, which is indicated by that duck and that large bump, and shows you that as viruses gain fitness, the global viral population in those animals can be extraordinary, and leading, in the case of influenza, to segmental reassortment, or the swapping of whole genetic segments, much different than the coronaviruses, but that can lead to huge leaps in the virus's capability, so that it might actually go from a reservoir species to a bridging species like the pigs indicated with the third surge. And lastly, if they get enough of a population there, they can have increased probability of going to humans. The point we're making is under the three statements underneath that. You must have surveillance to monitor these animals and see the inbound virus as it moves from wild birds to domestic ducks and chickens, for example, and that's your early warning. And you must look for opportunities to deflect those viruses by isolating the birds from, say, the pigs and other amplifying or bridge species. Potentially vaccinating your pigs might be an example. And last, you must prepare vaccines for threats that are coming your way. You have to decide at what point you're going to commit potentially $50 million to develop your first vaccine program. Okay, next slide, please. So what are the tools that you can use? Well, global research and new technologies such as our mathematics of virology. They'll look at the statistical algorithms such as that equation that's shown in this viral population diagram in the middle. This is actual research of viruses where the viruses were mixed in these evolution chambers, and we exposed those viruses to immune seros from humans, and we saw that the wild type, WT, was correctly suppressed down to almost zero and was eliminated from the viral pool. Now, what happened was very interesting. The remaining viruses that escaped the serum from a surviving human that was infected with that virus reordered, and that produced a dramatic increase in the diversity of the surviving strains. Restated, what this means we should be mindful of is the vaccines might exert an immune pressure that might lead to unintended consequences. While this has not been seen in human vaccinology, the new technologies are producing examples of this in animal models. So vaccines are an evolutionary pressure that viruses can try to get around, just like viruses can get around dengue with as you guys know, from antigen-dependent enhancement or antibody-dependent enhancement, and chikungunya with post-viral sequelae, such as arthralgias, okay? So the point we're making is understanding on a global scale, the vaccines need good scrutiny to make sure that we get the desired outcome and we minimize those escape mutants. So let's look at another example from a much larger project involving these technologies. Next slide, please. So here's an actual study that was done, and it was done by our colleagues in Jakarta, Indonesia. And what they found out is that if they used an H5N1 attenuated virus indicated by those blue polygons on the left, that's a living virus, by the way, it's attenuated. And they vaccinated their birds, their backyard poultry, indicated by those 6 chickens. What would happen is they lose a couple birds, actually. They lose less than 10%. But when those green birds were infected with a wild type H5N1, what would happen is the escape viruses that are shown on the right side of your slide were all the same. So the viruses that came out of the survivor chickens were very narrow. They were a single variant that emerged from those species. However, when that H5N1 pathogenic strain hit unvaccinated birds, they did not have this narrowing of selection, and the escape viruses in this study showed tremendous diversity. We, in fact, characterized with full virologic sequencing up to 9 pathogenic escape viruses from unvaccinated poultry. It's important to mention this might not be as big a problem as you think, because there's very few chickens that survive that infection. The lethality was about 96% in a 100,000 bird farm. So there was really several hundred chickens survived, but those chickens produce dramatically different viruses. So the observation, and this is not a conclusion, but the observation is that broad spectrum vaccines can reduce the diversity of variants. And maybe Li Fan and several of the other speakers will talk more about this with their pan-corona vaccine discussions. Our second point is this may decrease selection of very highly fit variants, and that could potentially lead to a reduction of subsequent waves in a pandemic. So there's a lot of excitement about this work, about using vaccines not just to protect yourself from infection and severe disease, but to prevent the escape of variants that defeat your vaccines. Next slide. Okay. So in the next 10 minutes, we're going to finish up in here and close in on several of the key points on the collaborations that we've learned about with the COVID-19 outbreak. So before we transition to that, we just want to summarize that pandemic-terminating vaccines are possible. We know this because we can differentiate between clinical immunity and sterile immunity. Clinical immunity is there is no disease, asymptomatic, but those people might be infected, versus sterile immunity where they are not infected. We can do sterilizing immunity with several of our more sophisticated vaccines and vaccines that go after multiple targets so that they're going to prevent the escape of variants. Okay, so very important point. Our new vaccines, we should always make sure that pandemic vaccines do not allow those vaccinated people to shed viruses, and that the immunity is directed against critical epitopes that are not prone to mutation. We call this less mutable. So the virus needs them, but they do not mutate. That's a great target for an antibody or a cellular mediated epitope. Thirdly, fast and inexpensive to manufacture vaccines will, pound for pound, kilo for kilo, do a remarkable public health service compared to waiting while your pandemic continues to escalate and produce many more casualties. So fast and inexpensive and easy to use are all the guiding criteria for the US's forward-looking vaccines. Reservoirs we talked about. If we can't immunize them with their own vaccines, we need to eliminate them, or we need to isolate them so we do not create a pool for the virus to go back and forth and to re-energize itself with new genetic material from which to create new surges. And lastly, we need to avoid opportunities for recombination, which is usually through co-infection. I raise this point because last week I was on service at my hospital in Boston, and I had an HIV patient who's on medicine, who's on and has a suppressed viral load, but still continued to express 3 separate COVID variants for greater than 21 days. Okay, so vaccinated person, boosted with a very good vaccine, in seemingly good health, on antiretrovirals with a very high CD4 count, greater than 400, and that person could not get rid of any of those 3 COVID-19 strains. Now, these strains from this single patient were not particularly virulent or sinister, but they posed a problem because they would continue to be in this patient and would be continue to evolve against that somewhat unusual immune system for that patient. So these people and these opportunities need to be very carefully scrutinized. Next slide, please. I'll end with a couple comments in the last 6 minutes of the importance of our pandemic collaborations. So I'm an American, and obviously the US is having a little bit of a problem with China right now, and China is having a problem with the US, but let us speak plainly. The US and the vaccine programs that we just told you about were dramatically accelerated by the relationships and the collegiality and trust of Chinese virologists and Chinese clinicians. And this is true in other countries around the world that might be politically difficult, even with Thailand, for example, but the scientists get along just fine. So it's important for us at this point of the pandemic to re-engage our partners internationally, to make sure that our collaborations are equal and fair and respectful, so that we continue to have everybody trained up, well-equipped, and ready to assist in the next pandemic. So with that, let's turn to just the following couple of slides. Next slide, please. Okay, so I use this slide because it's one of the first examples of an international collaboration where we were looking at the migration of also oseltamivir resistant and highly dangerous influenza strains, and this is H5N1. And if you look at those white, gray flyaways, you see all these interesting countries down from Indonesia through Cambodia, Laos, Myanmar, Russia, Northern China. They were all participating in the tracking of Tamiflu resistance because everybody would suffer if we had an H5N1 outbreak that defeated that widely available neuraminidase. And also on there, you see the amantadine resistance, which was predominantly tracking through Eastern China. So very important early example from way back in 2006. And it's gotten a lot better since then. More sequencing, more expertise, and a lot more data to share internationally. Next slide. So what does it look like right now? Is we now are understanding the importance of these collaborations, not only for the inventions that come out of some countries, but also our need to access patients and skilled clinicians and skilled basic virologists and research immunologists to help us understand these threats. Because far beyond COVID-19, it involves other diseases such as things that can really save us during influenza outbreaks. I'm pointing at the top slide, at the top picture of our friends at Balavet, an infectious disease referral hospital in Jakarta, who are doing boosted neuraminidase against neuraminidase-resistant viruses. It turns out that this is not an absolute resistance pattern, it is saturatable. So by boosting the neuraminidase much higher using different medicines, for example, cimetidine, which is very safe, we're able to improve the clinical cure in these patients. This could dramatically transform Asia's ability to fight bird flu outbreaks. In the middle one, we're showing some of the importance of several of the other ongoing coronavirus studies. This is a picture from a MERS outbreak in Riyadh and our partners there. And down at the bottom is me evacuating a patient who has a bacterial infection that's every bit as lethal as Ebola. It is a metalloproteinase VIN1, VIN2 resistant to every single antibiotic on the planet, as well as colistin. So that patient gets put into a very special aircraft to fly them back to a very special facility. So I'll just tell you that if you got a needle stick with an Ebola and you got a needle stick or were infected by that VIN2 carbapenemase resistant Klebsiella, you have the same case fatality rate. So we will see these bacterial resistance patterns in our daily practice in our hospitals in Bangkok and Hanoi and Hong Kong and Singapore. And we have to start really treating them based on the gravity of their infectivity and the futility of our therapies and look for new answers. With that is towards my second to last slide, and I think I'll end right on time, is that the growing, next slide please, is there's a growing need for us to really improve the collaborations. So I'll tell you what the humility of our partners in the US, those of us that work and live internationally, we have a very, we are helping our colleagues to understand the importance of these collaborations. And what the United States really needs to do, and what we are recommending our CDC and our USDA and our NIH do, is to energetically increase and reestablish our surveillance collaborations at our international partner sites and build human capacity, build careers there as well. The second point is to make sure we strengthen our relationships with our foreign laboratories that work with these dangerous pathogens, and so that we trust that work, because it's a laboratory just like our own. And Thai Red Cross would be an example of that. Hong Kong University would be an example of that. Nanjing Hospital, and certainly Duke and US are all superb labs that we've been working with for years. So strengthening that even further would help all of us. And lastly, to update our vaccine development tools to better assess whether our virus is natural or engineered. As we know, there is some controversy about the origins of COVID-19, and we won't get into that today, but let us accept the probability that many nations and pharmaceutical companies, while trying to produce ever more effective antivirals, will need challenge pathogens in which to test them against. And it is not unforeseeable that that actual environment could create more dangerous pathogens that, if they were to get out of these facilities, could cause a problem. Not saying that happened, but we do understand it's a product of biotechnology, R&D, and drug development. So we need to be mindful of that. And with that, I just want to turn to the very last slide, is that our global collaboration checklist must be based on trust. We got to play fair, we got to play equal, and we got to diversify our wins. So any gift that is found in one country should be spread for the public health advantage of all countries, because we're all in it together. The impact, we need to focus on the priorities of the countries and the people that we work with. If I come to, say, Laos, and I want to go after dengue, and that country is in the middle of a chikungunya outbreak, and they want to do chikungunya, I had better be doing chikungunya if I'm a good partner, and wait to get back to my dengue research. It's all about fulfilling the priorities of your partners, because you're a guest in their nation, all right? You need to help them with what they determine is most important. Sustainability is important. It's easy to start projects. It's hard to keep them going. And so one of the things that we focus on now is called not the startup, but the stay up. How you keep those labs moving after 2 years, with a goal of 5 to 7 years for self-sustainability, using host country support and getting them connected where they can go and secure their own resources. And the last thing is independence. It is our goal in these projects not to produce vaccines in one country and ship them around the world, but produce vaccines regionally at low cost, using the cheaper, more accessible technologies like mRNA. So each country will not have to wait for a vaccine to become available to it. That's fair, that's right, and we should all work towards that goal. That picture, by the way, was from the original SARS-1 outbreak, when we deployed at the request of the Hong Kong government in March of 2004 to Kowloon. So it's funny, it's been many years since that. It's been 16 years, and we're still working together with the same people. Those people helped us with the COVID-19 outbreak and understanding it. So it does work. And the last slide, the very last slide, is just, there's some additional reading for the references that go and support the points that were made in this talk. in the spirit of this discussion, in the spirit of the things we talk about, we speakers need to make ourselves available to mentor and guide, to make our materials available for your own education of young scientists and young physicians. So if I can help you as a personal note, my email is at the bottom, as is my WhatsApp. And so feel free to email me and just say that, you know, Dr. Chu and OPAS had sent you, and I'll make sure I answer your email. Thank you for your kind attention. And I'm sorry I couldn't be there in person, but I'll be back in Thailand in about a month and a half. Thank you.